All right, so we'll get started. I hope you're all refreshed from the very healthy juice. Great. I had the green one, so that's extremely healthy. So I'm James. So I'll tell you about. So this will be a tutorial. So it'll be hopefully an interactive tutorial, and the tutorial will be on optimal transport and applications of optimal transport to a few genomic examples. All right, so I think the timing is actually very good because just yesterday, one of the winners of the Fields Medal was actually one of the leading researchers in optimal transport. All right, so I think there's a lot of interest in the area. And the nice thing about optimal transport is I think it's one of the areas where there are lots of extremely interesting and deep, very active math. But the idea is actually extremely simple to explain to a broad audience, right? And in parallel, there's also been a lot of interesting developments and breakthroughs on the algorithm side, on the computer science side in the last few years. So it's right at the stage where I think a lot of these ideas are becoming extremely useful to a lot of computational biology applications. Right, so the goal here is to hopefully share with you some of the key ideas, not to get too bogged down in the math, but to share with you the key ideas and illustrate how you might use these ideas in some of your applications. So I'll illustrate the applications with 3 projects from my group. So to set the motivation, right? So here we're going to look at some single-cell RNA-seq data. Right, so oftentimes we're analyzing data sets, including single-cell RNA-seq. You often have the problem of how do you compare or integrate data from different experiments or from different batches. Right, so here each dot is one cell. Right, so each dot is a fairly high-dimensional vector of gene expressions for that one cell. And we have a bunch of dots from the smallest aging cell atlas from the pancreas, and have 2 colors here. So you have the red and blue, right? So the red and blue corresponds to 2 different batches of the same tissue, but 2 different batches of experiments. And oftentimes, what people have to do when they analyze this kind of data is they have to first somehow, you have data from different batches, you have to somehow put the data together. Right? And there's been a lot of computational methods very recently for how to put these batches together. Right, but the point is that after you put them together, you get something like this. And people usually either look at the PCA or t-SNE plots to see, is the data properly normalized? Are the cells properly aligned? Right. So the question that we want to ask is, when you look at these kind of plots, so how good is the actual alignment between these 2 data sets? Right? And how do you know if the alignment is actually good enough? So here's a different data set, different experiments, but also single-cell RNA-seq from a different tissue. And you can see that, OK, if you apply some of the recent methods that people developed, you also have visually a pretty good alignment. Right. OK, so here's the first question for you. If you are actually analyzing these kind of data, what you would do is that in practice, typically what people would do is you just look at these kind of plots, right, to visually see how good the alignments are. So who thinks that the picture on the, I guess, on the left here is actually a well-calibrated, normalized data set? Raise your hand. OK. Yeah, let's say the batch effect has been removed. OK, about half the people. OK, what about the one on the right? OK, a bit more people. OK, so it turns out that if you actually apply the standard metrics to compute how much batch effect is left in these 2 data sets, both data sets will actually be very good. Right, so this is one of the methods. You can basically compute essentially how much variance is remained after you do this normalization. Very good. OK. But I'm going to tell you a slightly different method for measuring how good alignment is. It's a very intuitive method, which is just that I'm going to simply treat this as a machine learning problem. Right, I'm going to train a classifier that takes as input each of these cells and simply predict whether each cell comes from red or blue. Right, so you take your process data, your aligned data, pass it through the classifier, and try to predict whether it's red and blue. Right. So I think most of you agree that if the classifier can actually predict very accurately, right, whether it's red and blue, there's still quite a bit of batch effect remaining. Okay. So the first data set Okay, so this is the classifier's, the discriminator's accuracy. So the data is balanced, right? So about 80% of the time, the classifier can actually classify correctly which cell it comes from, or which batch it comes from. And here's the second one, right? So almost 100% of the time, you can classify which batch it comes from. And this is after you've applied some of the recent alignment methods, right? So what's going on here? So even if the point is that if you look at this data visually, it's actually very hard to tell visually by how successful the alignment is, and in general, how different are these two data sets, right? But there could be a lot of high-dimensional correlations where the projections are not capturing. Yes.
I think you mentioned so you're learning your machine learning from a subset of data, and you're trying to learn the rest of that.
Yeah. So for my metric is going to be this discriminator that takes this input. So it's a standard machine learning problem, takes this input, a random set of these cells, so it's trained on a set of cells to train a classifier, and it's going to predict on the rest of the cells, can you predict whether it comes from red or blue? Yeah?
So most of the signals in lower species,
Well, so this is saying that if we just look at
trained on the first two PCs, we probably wouldn't have the same level of performance.
That's right. If I only train on the first two PCs, or if I only train on any low-dimensional projections, it's not going to be able to classify very accurately. But the data lives in thousands of effective dimensions, so it's just able to go. So there are still correlations that the standard normalization methods cannot capture. Right. So, okay. So, any other questions?
What's the name of that? The PVCA?
I think it's some. I can send you the reference, the principal variance component analysis, to capture how much variance is left. Yes?
So if you do t-SNE, that's still not separable?
That's right. Yeah, so visually, if you do t-SNE, you still can't visually tell them apart. Right? Which is problematic, because if you actually look at the papers that people publish, they always just show you the t-SNE plots, or the PCA plots to show you that the data looks reasonable. Right? Okay, so that's the motivation. So the general question that we're interested in is that if you have two data sets, first, how do you actually really quantitatively measure how different are these two data sets? And second is that what's the right way to think about how do you merge or align data sets? Right? And that's the scene for optimal transport. At the high level, the idea of the transport is that if I have two data sets, right, my red dots and my blue dots could be red cells and blue cells from two different batches. I want to learn a transformation so that I'm moving my red cells to my blue cells, but I want my transformation to be optimal in the sense that it's trying to move the cells as little as possible, right? It's trying to merge the two data sets while making the least amount of perturbations to individual data points. So just as a preview, so this is again, if I go back to this original pancreas data, if we actually do the optimal transport, I mean, visually, this is what happens. Visually, it doesn't look too different from the original data, right? But the point is that here, the discriminator cannot distinguish whether any classifier cannot distinguish whether you come, what's the batch of origin of individual cell. Right? And you can ask, okay, maybe that's because we didn't train a very good discriminator. So there's actually, the nice thing about the transport is that there's actually a theorem that guarantees that if you actually do the optimal transport, then no reasonable discriminator, no reasonable classifier, no matter how well you train them, can actually distinguish which batch of origin the cell comes from. Right? And I'll explain how that happens. But that's the idea, is that, okay, so in some sense, the optimal transport is, in some sense, the right thing to do. Okay, so that's the other cell. Okay, so if you do the transport, visually it doesn't look that different, but actually geometrically they are quite different. Okay, so here are the key ingredients of the tutorial. So the goal here, as we said, is that we have different data sets that I'm interested in comparing or interested in integrating, which is a very common problem in all sorts of computational biology. And what we're going to discuss here is this algorithm for combining data sets that we're calling optimal transport. And the algorithm will have both the algorithm and also a metric, right? The metric that the algorithm is trying to optimize. And the metric here will be what I'm going to define later. It's called the Earth mover distance. And I'll talk about two additional applications based on some works from my group. First is on using this idea of optimal transport to actually to do synthetic biology to generate new proteins. And the second one is actually use optimal transport to estimate site frequency spectrum, right? So it's very different applications, but hopefully to give you a flavor of why these ideas could be useful. Okay, so do a very dangerous thing to switch to the other monitor. Let's see. Okay, great. Okay, so I'm going to draw a few pictures just to illustrate what is the actual technical idea of the metric and of the algorithm. All right, so, okay. So just for simplicity, can you all see this? Yeah, okay. We're going to draw a bunch of points in one dimension, but everything can be generalized to higher dimensions. All right, so imagine if I have just two data sets in one dimension. I have a bunch of red dots, and I have a bunch of these black dots. All right, so that's my two data sets, and I'm interested in first defining what is a good metric, right, for saying how different are these two data sets. And then I'm going to talk about how do you actually transform one data set to the next one. Right? Okay, so what is a good metric? So the first thing I do is that, okay, so you have these bunch of data points, but equivalently, we can talk about somehow like the density or the distribution of these data points. Right? So these are equivalent. So comparing two data sets is equivalent to coming up with some metric for comparing two distributions, right? Two probability distributions. Okay, so if you're trying to compare these two distributions, right, the red one and the black one, I mean, what natural metrics would come to your mind? Okay, someone said KL. Okay, good. What else?
Distance between the means, some relation to the standard deviation.
Okay, good. Anything else?
Total variation.
Total variation, yeah. Okay, great. So let's look at those three metrics that people suggested and show why they may not be satisfactory. Right? So to illustrate that, let's actually look at even a simpler toy example. I'm just going to have the following three data sets or three distributions. So this is the density plot. So for the first data set, A, right, so all of the points are going to be just concentrated at location 1. Right. So this is 0, 1. This is B. Okay, so I have three data sets, right? Three distributions. So people suggested, I think, first suggestion is somehow like total variation, right? So total variation distance, also called L1 distance, right, between two distributions. Let's say in general, if I have distribution P and Q, it's defined to be the sum of I. Pi minus Qi. Okay, so it's very simple. So it just says, okay, if I look at these two distributions, just think of them as two vectors and take the distance, the absolute distance between these two vectors. Okay, so what is the total variation distance between A and B here? Okay, 1. What's total variation? Okay, so let me just define the notation. So the total variation distance is the same as the L1 distance, right? Because you're just computing the the L1 between these two vectors. Right, so what is the L1 distance between A and C? What is it? 1. Right. So this is a problem, right? Because intuitively we think of B as being closer to A, then C is closer to A, but the total variation distance, this L1 distance, actually gives them the same distance, 1, right?
Why is C closer to A and B?
Sorry, what's the question?
Why is C closer to A than B is?
They're the same distance by total variation, so 1.
So why is it like the other way? Why do you want B to be closer to A than C?
Because, well, intuitively, right, so B says that I have a bunch of points at Euclidean, at coordinate 2, right, and 2 is closer to 1 than 3 is closer to 1.
Oh, so 1, 2, 3 has a meaning.
Yeah, so it's on a real line. Okay. So the second one that people mentioned is KL. So KL is between two distributions P and Q is log of QI. Okay. So that's just the definition of KL. I mean, it's particular kinds of cross entropy term. So what is the KL between A and B? Okay, yeah, so KL between A and B is infinite, right? And the reason is that they have disjoint support, right? So because here you have some Q, Q here is B, right? So B1 is 0, so you're dividing by 0, and then you have all these things blow up. So KL is actually not even well defined for this simple distribution. From A and C, it's also infinite. Okay, so it's a very simple example, but it captures the idea that, okay, so the metrics that you think about, right, the standard metrics, maybe are not even for this very simple example, are not the appropriate metrics for defining distance between data sets or between distributions. Right, so the third idea that I think someone mentioned is, why don't we essentially fit a model to this, right? So maybe you can fit a Gaussian to the red dots and a Gaussian to the blue dots, and then you can look at the mean and the variance of these Gaussians, and maybe that gives you something. Right, so that's actually getting closer, but that depends on having to fit a very particular parametric model, which we're either unable or unwilling to do in general for high-dimensional data. Right, so how do I define a metric between any two arbitrary distributions that captures this appropriate notion without actually having to define a parametric model? So that's the third option. Is called the Earth mover distance. So the Earth mover distance, let me give you the intuition first, and then I'll write down the mathematical definition. The Earth mover distance is basically saying, if I'm trying to, between two distributions, right, P and Q, is to say, how much do I have to pay to move from P to Q? Right, so let me be a little bit more precise. Right, so imagine if you're a moving company, and you're thinking of, you have to move from A to B, right? And you have a bunch of stuff, a bunch of furniture at A, and you have to move all those furniture to B, right? So how much do you have to pay as a moving company? Well, then you have to pay by how much furniture you have to transport and the distance of transportation, right? So the Earth mover distance is basically somehow like the mass you have to move times the cost of moving, right, or the distance moved, which is some notion of cost. So for this example, the Earth mover distance between A and B is 1, right? Because I'm taking the masses here is 1, and I've moved distance 1 to go from A to B. The Earth mover distance between A and C is 2. Right? Take 1 unit of mass, I have to move it to distance of 2. Right. So the Earth mover distance, it's a valid distance over distributions, seem to capture these more fine-grained distinctions between different distributions. Any questions about this? Okay, so that's the metric, that's the Earth mover distance, right? So optimal transport is the algorithm for minimizing the Earth mover distance. So let me explain what optimal transport is. Yes?
Isn't this very sensitive to any like change of coordinates or like any transformation that you somehow do on your data?
So it depends on what is the notion of distance, right? So that's something that you do have to specify ahead of time. Yes? Okay, so how do I actually, so all I've given you is giving you a definition, right? So how do we actually algorithmically solve this problem? So the algorithm, optimal transport. Okay, so let me, I'll write down a couple of equations, and then I'll parse it for you. Right, so basically the algorithm is that the Earth mover distance between two distributions comes from solving a particular optimization problem. Okay, so don't worry about what's going on here so much. So basically, the idea here is that T is going to be my transport, right? The transport is going to be how much your moving company is going to, which route it's going to take, right? So we're going to try to find the best moving company, so we're going to try to find the optimal transport. So what is a good transport? The transport is one that minimizes the total cost. The total cost here is that, so we sum over all the points I and j, right? So the first term is what's the, how much stuff are you going to move from point I to point j? And the second term is the cost, the distance of moving from I to j. Right? Does that make sense? So Cij is the distance, it's the cost. Right? This is C here. And Tij is the transport. How much are you going to move from I to j?
So in your example, Cij is just I minus j? Or
Exactly, yeah, exactly. And this stuff is just a fancy way to write a very simple intuition. It's just a conservation of mass, right? So I want to say that my moving company is going to move, however it's going to move it, has to move exactly pi items from position I. And after it moves it, then there has to be exactly qj position, items left at location j, right? It has to move from here to here. Okay. So this is the precise this is the precise, in some sense, the precise definition for what Earth mover distance is. But it's also an operational algorithm because this is just it's called a linear program. The inputs are linear program was the input is the matrix C, P, and Q, and the output is this T, is the transport. So the point is that the linear programs are something that we can solve exactly, and we can find the global optimal solutions efficiently. Okay. Any questions about this? Okay. So this is what I want to say. This is basically the primary part about what optimal transport is, right? So the key takeaway message is that optimal transport has basically two ingredients. The first is that there's a definition called Earth mover distance, which in many cases is the appropriate notion of the appropriate metric to compare different data sets or compare different distributions. It's the first component. And the second component is that it actually also has an algorithm attached to that definition, and the algorithm is it's something that we can solve. Okay, so just to
On the side, that way?
so I'll draw the picture again. So if you didn't remember all the details, but the picture to keep in mind is that you have your, you have some distribution P, and you have some other distribution Q, right? T is the transport, and you're essentially, you think of each distribution as being some point cloud, right? Like in my single cell example from the beginning, the cells is actually literally a point cloud. I'm trying to learn a mapping from the first point cloud P to the second point cloud Q, so that the distance, the amount of work done is actually minimal, right? So that's my optimal transport. Yes?
This is a super nice question, but so like in this case, this works either if the two distributions have exact same number of points, or you need to kind of put a probability distribution on top of it with some smoothing, or how do you apply this? Or more
Yes.
to apply this if the total number of points varies.
Yeah, this is a good question. So if they have different number of points, then we just normalize them into, divide by the number of points into, so that they become a distribution.
Split your chair into two half chairs.
That's right. Yeah. Okay, good. Okay, so now I'll talk about two applications of this. So the first one is on applying this idea to, well, so we have already seen one application is to the single cell case, right? So the transport that I did there to get the optimal, to get the best alignment is exactly from applying this optimal transport. So the second application I want to show you is on how do you actually apply this idea to, to generate new, maybe interesting proteins, right? So,
This falls on Dan. If you had the same number of points,
yeah.
they would have gotten identical. After you applied optimal transport, the two data sets would have been identical.
The data sets would be identical, yes. Yeah. Okay, so let me draw a picture before I go on. So the idea here is that, right, so the transport can map any distribution P to any distribution Q. So in our case, let's think of Q as being some interesting distribution that we're interested in from a biological perspective, right? So it could be the space of different proteins that satisfies a particular biophysical property, some desirable property. And maybe I don't know explicitly what that distribution is, but I can go on different databases and can find examples of proteins that have that property, right? So I have some point cloud in some very complicated protein space. Okay, so how do I, what's one approach to try to model that distribution of protein space and to generate new proteins? So it's a very naive idea, which is, okay, so let's start with a very simple distribution that I know, which is just Gaussians, right? So, a spherical Gaussian. So this is my proteins, Q. So I have some P, which is just random Gaussian. And I'm just going to learn a mapping from the Gaussians into this protein space, right? Learn a transport. Right, so the reason for doing that is that, so it's easy for me to sample from Gaussians, right? And if I have a good mapping, then I can just sample from my Gaussians and then apply the transport to it to potentially find, maybe it will transport me to new points in this nebulous protein space that I haven't seen before. All right, so that only works if the transport is, it's not too bad. So in machine learning, this is basically what people have been calling sort of generative adversarial networks. It's usually presented in different ways, but essentially it's trying to solve this problem of how do you learn transports from a very simple distribution like Gaussians into a more complicated distribution like the distribution of images, or in our case, the distribution of proteins that have some desirable property. Right. but it is using the same formulation that we described on the previous slide. Okay, so now let me switch back to my laptop. Okay, so the first application is so this application is on generating new proteins using exactly this approach. Right? So there's a little bit more bells and whistles to it because we have to somehow not just generate proteins that we've seen before, but maybe new proteins that have even better properties than the ones that are currently feasible. Right? And the motivation for doing this is that if you're thinking about exploring the space of possible proteins with some desirable physical properties, like the ones that we have empirically observed maybe only make up a very small fraction of that total space. Right? So we want to be able to efficiently sample and to model that entire space. So this is work of a very talented student, Vita Gupta. So what we did is that, so we applied this approach, right, this transport approach, to generate proteins with different types of desirable properties. But the first thing we want to show is that this very naive idea of trying to map Gaussians into some complicated protein space actually works. Right? You can actually get things that look sensible. So one way to test this is that just naively, right? So I have some family of proteins that have just their distribution of amino acid usage, right, of the real proteins in that family. And I just want to see that of the proteins that I generate with this generative model, do they have similar distributions? Okay, so it turns out that they do have similar distributions. But the amino acid usage is a very crude metric, right? So then there are lots of other metrics that one can use to measure different physiochemical properties of these proteins, right? So here's basically basically you take the 10 most common types of biophysical properties of proteins and then just do a PCA plot of them, right? So each dot here in gray is like one real protein from a desirable family, plotted along those different physical properties. And each of the red cross is one of the synthetically generated proteins just from this transport. Right. So they also have, at least by these simple metrics of the properties, they have also reasonable properties. And the nice thing is that if you actually compute what is the distance, right, the edit distance between the proteins that are being generated and any of the previously observed proteins, they're actually quite far apart in the distance space, right, in the edit distance. So they're not generating, just memorizing and generating existing proteins. Okay, so the real test is, can we actually synthesize these proteins and actually functionally test these new proteins, whether they have interesting properties? Right, so that's something that's still very much ongoing work. So here's just a very simple proof of concept, right? So can I first just generate proteins that have certain types of desirable secondary structures, right? So here we basically want the transport to generate proteins that have basically different types of helices, right? In particular, we want to have helices of certain length, right? So so the red distribution corresponds to what is the distribution of helices before you apply this transport, and then after you do the transport, you can see that you're getting proteins that have much longer helices. Right? Here are some of the ones that we have generated. So the second proof of concept is, can we generate proteins that have desirable, like maybe antimicrobial properties, right? And again, we have to do much more extensive functional validations of this, but at least just here's the histogram shows that the blue one corresponds to the distribution of proteins before you really apply the transport, and the red one corresponds to after you optimize and apply the transport, at least by bioinformatic approaches where we try to measure the antimicrobial properties of these different peptides. After you do the transport, you're generating peptides that have much greater antimicrobial properties. Again, needs to be validated. Yes?
When you're saying before, you mean when it's just random noise?
When it's basically just generating random proteins, yeah. Yes?
What's the notion of box here? Because the transport sounds like it's one step, but
Yeah. So that's a good question. So basically, the transport is solving an optimization problem that optimization problem I didn't explain in too much detail. So the optimization problem basically can be solved iteratively in a few different steps. So the epoch here tracks how many steps of optimization have we done.
This is for the linear program?
So this is actually for something extension of the linear program. So basically, the linear program works in general, except in settings where we have continuous distributions. Then things are not finite anymore. So then there has to be a continuous extension of the linear program. So that's worked out mathematically, and then this is what we're doing operationally. But the idea is basically exactly the same as a linear program. OK. Oh, yeah. So in the remaining 5 minutes, I'll show you one other application, a very different domain, which is actually more related to PopGen, which is estimating variant frequency. Right. So the setup here is that if this is my entire population, right, if my entire population consists of these 10 individuals, and each column corresponds to one variant, then what I can do is just for each variant, if I have actually sequenced everyone, I can compute what is the frequency of each variant in my entire population, right? And the histogram of that frequency counts is also what people call sort of like a side frequency spectrum, right? So that is what you have, right? So that's something that I would like to estimate, right? Because in practice, I don't observe this entire distribution of individuals. I only have a finite number of random samples from my whole population, and maybe I only have 5 random individuals. Right? So then what I can do in practice is that I can compute among the 5 individuals that I've sequenced, right, what is the empirical frequency at each variant. Right? And I can compute my empirical side frequency spectrum. Right? So it's a very fundamental concept from population genetics, and if you can have this distribution, this histogram, the side frequency spectrum, you can do a lot of interesting things. But the thing that we're interested in here is that, how different is the empirically observed side frequency spectrum from the true distribution, right? The true distribution that I would have if I have the entire population. So they could be very different. So in particular, for the variants that are more common in the population, then the empirical one will be very similar to the true one because those variants are observed very frequently. But especially for the rare variants, right, my empirical SFS is going to be very different from my true distribution, right? And again, here the goal is that I now have 2 distributions, right? There are over frequencies of variance, right? I'm going to define the metric, the Earth mover distance between these 2 distributions, and I'm going to formulate this as a transport problem, try to take my my observed empirical frequency and try to estimate the true frequency, right? And we've done this with various data from EXAC, and I won't describe the algorithm in too much detail. It's the same linear, it's a related linear program to the one that I described before, right? Trying to optimize this transport problem, but it's again a linear program, which means that we can solve it and we can find the global optimal efficiently. And here, the theoretical guarantee is that if I solve this transport problem, if I solve this linear program, then I have a guarantee on how good my estimate is. Right? So the left-hand term corresponds to what is the actual Earth mover distance between my estimate of the side frequency spectrum and the true population frequency spectrum. Right? The theorem that we can show is that that distance, the Earth mover distance, is always bounded and it's always decreasing very rapidly as I sequence more individuals. And the nice thing here is that we can actually show that this is that doing the transport here is actually the optimal algorithm, right? So you cannot actually achieve a faster decrease in the error rates with any other algorithm. So the details here are not so important, but the main idea is that you can apply this kind of transport and this Earth mover distance even in the case of this population genetics. And we've done other applications, follow-up of this, to use this to estimate, to do various extrapolations. But I just want to maybe wrap up with a discussion here, right? So I think the key technical messages I hope to convey with this tutorial is that if you have different data sets, oftentimes I think you have to be very careful about what is the right way to measure the similarities and distances between different data sets. Right? Standard metrics like KL or visual inspection are not necessarily the best metrics. In many cases, Earth mover distance, I think, is the appropriate metric. And optimal transport is the algorithm that achieves optimal Earth mover distance. And there's a lot of interesting applications that we've done in single-cell RNA-seq, proteins, SFS. But I think we're just scratching the surface of what could be applications of this family of techniques to computational biology. So I thank my students, and especially Rishan here. She's standing right here, who's done a lot of the work that I described in the paper, in the projects. I'll take more questions. Yeah.
Linking this arsenic distance back to the batch effect detecting example, how do you grade that? Like whether there's batch effect according to the arsenic distance is so large distance means there's clear difference between those two distributions?
Yeah, so great. So that's actually one thing I did not have time to mention. So it turns out that, right, so here, so the Earth mover distance is actually here the same as the discriminator's performance. So there's a mathematical theorem that I didn't have time to state that says that the Earth mover distance between two distributions under some mild conditions is the same as what is the performance of a discriminator, the accuracy of the discriminator in classifying the origin of this distribution. Right? So the discriminator here, so this is basically the same as the Earth mover distance that we're computing. Right, so there's a very natural duality between doing this classification problem and computing the Earth mover distance. Yes?
So it's a great talk. I didn't fully understand when you gave the side frequency spectrum example, or when the target distribution is unknown as to how you model the problem. You find a distribution that's closest in some sense, but you don't have any other information other than what you have on the empirical distribution.
Yeah. So that's a good question. I didn't explain that part. The algorithm I glossed over the algorithm. So the algorithm is that basically we're going to take the observed empirical SFS, and then we're going to construct a new distribution to basically match the moments of the empirical SFS. But the new distribution, that's my transport, is going to be constructed in such a way that it's going to minimize the transport cost from to the true distribution. So that's exactly this linear program that I glossed over, but I'm happy to go over that afterward.
So for the single cell RNA-seq example, I guess I'm wondering, I mean, it seems like this metric of the discriminator performance, there can be like trivial ways to get perfect discrimination where you basically just match two data sets to be exactly equal, but that could like potentially overfit in some cases. You don't want to align
That's right.
that well. So how do you, I don't know if the optimal prevents that.
Yeah.
And do you have some independent like validation to make sure you're not like over-aligning through this?
Exactly. Yeah. So both are very good points. So the intuition of the optimal transport is exactly to prevent that kind of overfitting, right? Because the idea here is that since you get penalized for moving things too far away, right? So you're encouraged to make only local changes, right? So then you can't just take these points and then randomly put them to match up exactly with your second data set. And we do basically something similar to having a holdout data set and then evaluate the performance in the holdout data sets. So the transport is it's much less prone to overfitting than other approaches. Yes?
So this seems to be an example where all of the differences between the groups is unwanted variation. Can you say anything about the case where the batch variable is confounded with a variable?
Yeah, that's a great question. So even in this example here, right, because you actually have potentially different subpopulations even within this tissue, and that's the case in general for a lot of single-cell RNA-seq data. You have particular subpopulations, and you don't want to necessarily match them all together nilly-willy, right? So what we actually use is a little bit, is a new algorithm that we developed that we call hierarchical optimal transport, which is basically first identify subpopulations and then only transport the matched subpopulations to each other. So here you're right. So if we just apply the whole optimal transport to this entire thing, it's going to basically crush all of the technical variation, but it's also going to crush biological variation across the data sets. Yeah.
For the distances in the distance part of that algorithm, is it adjusted so that if you had, say, strictly integer value data, that you would restrict the amount that can move in time to be integer jumps versus, say, real value data? Because I think when people are using other batch correction methods, sometimes they ignore that kind of the domain of the data when they're trying to do bring together batches, for example.
Yeah, so I think if you define the cost function, which is equivalent to the distance in particular ways, you could ensure that the transport would have either real values or particular integer values.